Hello! This is our Metabolism project by Ainsley Buia, Buia Matai, Kennedy Thornton, and Annie Wineka. The first step of cellular respiration is glycolysis. This process occurs in the cytoplasm of both prokaryotic and eukaryotic cells, and in as little as 2 phases, the system breaks down glucose into pyruvate and stores the energy from the bonds in NADH and ATP. So in the cytoplasm, this is called the investment phase. 4 ATP go into the bonds of a glucose or 6-carbon sugar to break the bonds and produce 2 pyruvate molecules or 3-carbon sugars, and the energy from the bond breaking converts 2 ADP molecules into ATP molecules and 2 NAD plus molecules into NADH. So it reduces both low energy electron carriers into high energy. The next part of cellular respiration is the link reaction. This kind of flows between glycolysis and the Krebs cycle. This reaction occurs in the mitochondrial matrix of the mitochondria of a cell, and this only happens in eukaryotic cells. This reaction turns pyruvate into acetyl-CoA to then give to the Krebs cycle, basically. All right, in the mitochondrial matrix, the pyruvate molecule, this is the pyruvate molecule, and the coenzyme A attaches to 2 of the carbons in this molecule and kind of pulls them away from that 3rd molecule right there, which produces 1 molecule of CO2 gas and 1 molecule of acetyl-CoA. And the potential energy that is in that chemical bond that just broke reduced 1 NAD plus molecule into NADH, and that is per pyruvate molecule. So per glucose molecule, because there's 2 pyruvates in each glucose molecule, there will be twice as much happening. So 2 molecules of CO2, 2 acetyl-CoA molecules, and 2 reductions happening.
So then for the Krebs cycle is the last step in cellular respiration. It's a cycle that produces the rest of the carbon dioxide from the pyruvate. The Krebs cycle or the citric acid cycle occurs in the mitochondrial matrix, and it occurs twice for every twice because there's 2 pyruvate molecules. So it starts with the acetyl-CoA from the link reaction, and then because it already did its job in the past cycle, it then leaves. And then the oxaloacetate bonds then bond together to create citrate with the pyruvate. And then the bonds break from the oxaloacetate to create 2 carbon dioxides, and then it continues to do it with the other pyruvate molecule.
The electron transport chain is the last step in aerobic respiration and the first step in oxidative phosphorylation. It happens in the mitochondrial membrane in eukaryotes and the plasma membrane in prokaryotes, and it also happens in 4 different complexes. So the electron transport chain begins with the electron carriers NAD plus or FAD, and they deliver the electrons to the first complex. Then they move through the next 3 complexes. And as these processes occur, enough energy is generated to pull hydrogen ions through the membrane, leaving the mitochondrial matrix and into the intermembrane space. And at the last complex, an electron, like O2, grabs the electrons and forms a molecule of water. And the electrons needed for glycolysis and the link reaction are refueled from NADH into NAD plus and FADH2 into FAD. Photosynthesis is the process used by photoautotrophs to convert an energy source into usable energy, and it occurs within the light-independent and light-dependent stages. So in the light-dependent reactions, photosystem 2 breaks down H2O, which pumps hydrogen ions into the lumen. Light strikes photosystem 2, causing the excitement of electrons, and these electrons move through the electron transport chain to photosystem 1, causing hydrogen ions to be pumped into the lumen. Light then strikes photosystem 1. This causes electrons to get excited and move to the NADP plus reductase, and this fuels the formation of NADPH from NADP plus. The NADP plus is reduced into NADPH.
Okay, so the Calvin cycle is the second component of photosynthesis. It is also known as the light-independent reaction, but the cycle actually is dependent on the electron acceptors produced by the first step of photosynthesis, which is dependent on sunlight. So carbon fixation occurs when 6 carbon dioxide molecules bond with 6 RUBP, and the reaction is catalyzed by the enzyme rubisco to form 12-PGA. Okay, so ATP and NADPH is then oxidized from the light reaction and comes into the Calvin cycle. Then ADP and NADP plus return to the light reaction. This leads to the reduction where the PGA is reduced to form G3P, and it forms 12 of them. 2 of the G3P are used to create 1 glucose molecule. The remaining 10 G3P molecules are regenerated into 6 RUBP in order to begin a new cycle. 1 G3P molecule is used to make glucose, while the other 5 are recycled, and then the cycle continues.
Which takes us to chemiosmosis. And chemiosmosis is the second step in oxidative phosphorylation. And unlike the electron transport chain, this happens in both photosynthesis and cellular respiration. And this is all kind of centered around an integral protein called ATP synthase, which is an enzyme that is built into the inner mitochondrial membrane. And chemiosmosis is responsible for making a lot of the ATP in the whole system that we have going on here. So it makes around 30 to 36 ATP. So we have our wonderful little ATP synthase in our membrane, and it helps the hydrogen ions move down through their gradient. And the movement of hydrogen through the ATP synthase enzyme rotates the enzyme, and this pulls ADP and phosphate groups together, and they clash inside. And this mechanical energy causes them to bind together, and this creates a lot of ATP, as you can see by our wonderful design. And this happens again and again and again until it makes a huge amount of ATP, which is why it counts for a lot of the ATP made in the metabolism. Chilling.